guess ask you both of you about discussion with, for example, designing robots at open-ended environment. And that's something I think in biology how these creatures already figure out to do that, to be adaptable to uncertain situation. So when it comes to the discussion between both of you, I'm curious how for you as a computer science and computer science, how you can design system, living system that can work at open-ended environment, scenario like that. What are the challenge? What are the question? Do you both of you share and consider maybe?
Yeah, I think that's a great question. How to reconcile the open-ended environment. Um, so just for your listeners, maybe it's helpful to set up the Xenobots project where we use an evolutionary algorithm and run it in a supercomputer to design artificial biobots in a simulated environment, and then Mike's group tries to instantiate it in biological tissues. So the immediate question we have to ask on the simulation side is, what do we put in to the physics engine? What aspects of biology and physics are is living systems exploiting? How do they exploit it? And how might biobots exploit it to do what we want it to do? So
Mm-hmm.
turn over the floor to Mike and that.
Yeah, I mean, one of the important aspects for us is to understand the natural plasticity of the system itself. So one of the amazing things about the Xenobots is that we take cells with a completely normal wild type genome, meaning no genomic editing. We don't change the cells in any way. And we're finding that they, as a collective, are capable of doing some really novel things that they otherwise normally don't do. And so, for me, part of this whole adventure is to figure out ways to computationally explore and predict the space of possible outcomes that we're normally not used to. So, in biology, and in particular in developmental biology, we are often this plasticity is masked by the reliability of development, right? The fact that a frog egg, if you don't do anything else to it, very reliably makes a tadpole. And so you sort of, as certainly students, sort of get this feeling that, okay, this is what this machine is able to do. But what we're seeing now, and of course people have seen aspects of this before with in terms of epigenetics and other types of experiments, that actually no, these cells have very much greater capacities for doing things both collectively and singly than what they normally do. And so how do we learn to predict that? How do we learn to anticipate the kinds of plasticity that these cells will have? And so here I think is the grand challenge, is to use the computational tools to do that.
And I think one of the interesting things about Mike's work that we find so fascinating on the computational side is viewing the organism at the cellular level. That in a way the organism is made up of intelligent machines at the tissue level, the cellular level, the subcellular level. So in many ways, for different parts of the xenobot or for different parts of the organism, the open-ended environment is the cellular environment itself, is the neighboring cells and the signals that are coming in and being sent out. So when the evolutionary algorithm attempts to rearrange simulated biological tissue, then one hunk of biological tissue now has a new environment. It's up against a new piece of tissue that may be in the adult frog, the wild type frog. It hasn't been in contact before. What do we know about that novel rearrangement? How do we simulate it? Do we put noise into the simulator? How do we ensure that that novel rearrangement will succeed if we try and build it in reality? That's really kind of an open-ended question for us at the moment.
Yeah, so on the biology side, the question of learning is fairly contentious, because some people take learning to be a very narrow sort of thing that advanced animals with brains do, and then there's the field of basal cognition, which sort of stretches it out into a more, I would say, evolutionarily realistic view, where learning shows up very early on in the web of life. And so um, the thing about the interesting thing about learning is that you have to have a subject that learns something, right? And you have to when you have memories and learning, it has to belong to some agent that you have to be able to describe. And this is very non-obvious, because all cognitive agents are made of parts. So when you have something that learned, you have a bunch of it. So if a rat has learned something, what you really have is a collection of cells. And so let's say the rat has learned that when it presses on a particular lever with its foot, it gets some apple juice or something as a reward. The cells that interact with the lever are not at all the cells that get the reward. And so the trick then is, and so when you say the rat has learned something, well, what's the rat? The rat is some collective of all of these cells that have figured out how to do credit assignment properly. And this, of course, is very familiar to machine learning and robotics folks. Is that the trick here is to understand how all of the individual cells have bound together into a larger agent that is the subject of rewards and punishments and things like this. Very non-obvious. This also works in individual cells. So single cells can learn, and plants can learn, and things without brains. So it's not just a story about brains and neurons. It's a story about how competent subunits can work together in a way that gives rise to a larger scale self, that it can be the owner of memories, the center of preferences, the subject of past experiences, and so on, that are not individually true of any of the components. So that is one of the grand mysteries in biology, and so we're just starting to really understand how that works in the global sense.
Yeah, I think, there's a lot of brand new questions, I think, for the AI community now that sort of biohybrid technologies are becoming possible. So, intelligent prosthetics, biohybrids, if you put technology together with living systems, the living system usually learns, right? It will learn to send the right signals to the prosthetic and receive the signals appropriately. Again, because living systems are so good at learning, and as Mike said, have solved the credit assignment problem, they're more or less willing to work alongside technology and share the credit for collective action. But it's the prosthetic itself, the artificial side, that still grapples with learning how to interpret the signals that are being sent from the living side of the biohybrid. So I think, there's some brand new kind of machine learning problems you can ask at that level. There's another way to connect technology with living systems, which is what Mike and I have been doing recently, where the learning is the evolutionary algorithm that's learning how to rearrange biological tissues into new forms and functions. And that's, that's quite different from biohybrids, where you're marrying hardware and robotics with part of an organism. So what are the questions there if you want an evolutionary algorithm to learn what rearrangements work and what rearrangements don't work? That again is sort of a brand new way of formulating, a question in AI. And lots to do.
Yeah, so there are a number of organisms that are able to repair themselves after damage. And so axolotls, the Mexican salamander, is one example where they will regenerate their legs, their eyes, their jaws, portions of the brain, the heart, their spinal cord. There are other models like the planarian flatworms that we work with, where pretty much every piece of the planarian can regrow the entire thing. So this ability to regenerate is a special case of a broader capacity, which is something we call anatomical homeostasis. So this is not just for adult regeneration. Embryonic development does the same thing, and so does remodeling, for example, in metamorphosis from a tadpole to a frog. The idea is pretty simple. You have this homeostatic loop that continuously deforms and remodels the tissue, so the cells are moving around, they're migrating, they're differentiating, they're doing various things, until the shape becomes roughly equal to a target morphology, the shape that it's trying to achieve. And so it's basically an error minimization scheme that it's able to, in some coarse-grained way, sense deviations from the correct pattern. And those deviations could be injury, they could be teratogens, they could be mutations, they could be all sorts of things. And so, and until that error is reduced below an acceptable threshold, things will keep changing. So for us, the real trick is to understand not only how regeneration happens, and lots of people study the cellular mechanisms of turning this cell type into that cell type and so on, but actually the algorithms of how it fulfills the 3 steps of that homeostatic cycle, because it needs to do 3 things. It needs to measure the current shape so that it can tell whether it's correct or not. So it has to, so the collective, and this is something very large, so no individual cell can measure something so big. It has to be a collective computation. So the collective has to be able to measure the current shape. Then it has to be able to remember what the correct shape is. So there's a kind of pattern memory here that where you can tell, is it right or is it wrong? And we've made some progress in seeing how it stores those memories. And then it has to issue the collective has to issue commands to the lower level subunits, meaning the cells, to rearrange in a way that gets them closer to this final. So to us, this is very much this kind of problem solving cycle, really critical to find out how does it know when to stop. Very few people actually work on regeneration, asking how does it know when to stop. So when a salamander makes a correct arm and then everything stops, how does it know that's what a correct salamander arm looks like? I mean, that's a critical question. And so all of the three, sort of all of the aspects of that standard sort of test, operate, exit loop has to be identified in biology, and we've really just begun to do that.
Mm-hmm. I'm curious to ask you, just based on what mentioned, do you think in robotics we have to go for this question just answer them, or it doesn't necessarily to go to this level?
I don't know about this level, but absolutely a lot of what Mike just said has to be incorporated into robotics. We're moving in robotics into soft robotic spaces, into biohybrid robots. There's been this assumption throughout the history of robotics that the body of the machine is fixed. And that assumption, has to be rolled back. You cannot, in my opinion, you cannot make an adaptive machine that has a completely fixed body plan. It just doesn't work. You're just too limited. But again, if we're building, depending on what soft robots we're building, out of what materials, we can't just borrow directly from biology, right? We couldn't make a huge flapping bird and get it off the ground. We had to borrow the principles of heavier than air flight and adapt it to the materials from which it's built. So I think, as usual in bio-inspired AI and artificial life, we're looking carefully at what Mike's group is discovering and figuring out how to distill out of that the basic principles of homeostasis, adaptation, intelligent action, and then build that into whatever materials we're building our soft robots out. And how to do that well or in a principled manner is, again, a completely open question.
I mean, the funny thing is, it's interesting that you say that about giving up that assumption of a constant body. Biological evolution gave this up long ago, because the interesting thing about many, probably not all, but most creatures is that they are incredibly tolerant to large scale changes of architecture. So, for example, you can make we've made tadpoles where the eyes are on the tail, and those instead of in the head, and those animals can see perfectly well. They learn in visual assays, no problem. So the brain can immediately, well, first of all, at the cellular scale, if you are a bunch of cells that are trying to make an eye, the fact that you're sitting next to a spinal cord and muscle as opposed to the brain and various other things, no problem. You'll still make an eye, and you'll still make an optic nerve that connects to something, the spinal cord nearby, perhaps. And so that plasticity is there, and then the plasticity of behavior. So the brain has no trouble interpreting this weird, sort of electrical signals from a new patch of itchy tissue on your tail as visual data and behaving appropriately. It handles that right off. And so what that is probably signifying is that evolution discovered really early on that in order to be adaptively successful and evolvable, you have to have that kind of plasticity, that you have to assume that your body can change, and that things don't necessarily go where you can't assume that everything is where it needs to be, and that you can still all the different parts compete and cooperate within the body to result in some sort of adaptive function. So that plasticity, I think biology sort of assumes right off the bat that you don't know where anything is, and that you still have to get your business done, even if things are surprising and not where you thought they were.
Mm-hmm.